Okay, so can you see the slide now?
Yes.
Okay, all right. So, so the subject today is going to be on microbial bioremediation, and so the reason is, in concept, remediation has exactly the same meaning, except that here we are going to look at how microbes are useful for the for the process. And essentially, the kind of chemicals that we are going to be looking at when you talk about microbial bioremediation are the chemicals which are released from various industries, including, the agricultural practices. So these are the ones which are actually responsible for adding a lot of pollutant load in the environment. Now, when we started to discuss about environmental biotechnology, I also mentioned that generally we say there are non-point sources and there are point sources. Point sources are the ones which are very close to the industry. So just at the site itself, as a byproduct of industrial activities, several pollutants are released into the environment, either as an effluent or it could be a soil, it could be a solid waste like you saw in the case of thermal power plants. And these are called as the point sources of pollution. And in this, one of the major polluting industries is all the energy producing factories. For example, when you process coal in a thermal power plant, or when you are going to process oil for production of energy, it could be diesel, or it could also be the crude oil which you are going to process to get the fine petrol, or it could be the nuclear resources. All of these will get classified under energy producing industries. Okay, so coal processing, oil processing, nuclear processing, etc., we say they are all energy producing industry. And so these are, they add to, for example, to the point sources of pollution. According to US environmental protection agencies, there are about a million, because the data is only available for the US, but I'm sure even in India and in several other countries also, there are underground storage tanks which store gasoline. Okay, because once once the oil is refined and through various pipelines, it is sent to underground storage tanks, like in a petrol pump or whatever, you do have underground storage tanks. And in US, it was estimated that there are about a million such underground storage tanks. And out of which, what has been recorded is that since 2013, there have been about 500,000 cases which are reported of the leakage, which is causing hydrocarbon contamination. So over a point of time, many of these tanks, because of overfilling or because of cracks in the earth, the petrol, it starts to leak out into the adjacent soil. All right, and the cases which contribute are extremely large. So this is something which you call as hydrocarbon contamination. So once the petrol gets out into the adjacent soil from these storage tanks, the soil there becomes contaminated to a large extent. Okay, now amongst the non-point sources, okay, so non-point sources are the ones that you are using an industrial chemical at some particular site. All right, but then eventually, because of either floods or because of rain, these pollutants, they get carried over large distances. So they end up at a point which is not necessarily where they have been used. That is called as a non-point source of pollution. And agricultural industry is one of the major contributors to this non-point source of pollution. What kind of chemicals are used there? So if I am going too fast, if you want me to slow down, so you please interrupt me. Okay, we have
No,
already
it's
lost
fine.
some time. Okay, so I'm just going a little bit fast, but please interrupt me. So in the agricultural industry, a lot of these pesticides, fertilizers, they are applied over very large areas. And it is not something, which is just done once in a while, but, food is grown all through the year, and large areas are under agricultural land. And if there is a rain, okay, then the pesticides and the fertilizers, they get carried over very large areas. And what has been found that more than 90% of the monitored streams and 55% shallow groundwater. So when they get carried by rain, the groundwater, which is, water which is just below the subsurface of the soil. That is called as the groundwater. And it has been estimated that about 55% of this groundwater gets contaminated because of these pesticides, fertilizers, which are released through agricultural practices. Okay, now what is the problem with groundwater contamination? Number one, if the groundwater gets contaminated, okay, this makes up 97% of the available fresh water which is supplied for drinking purposes. So the water which is called as the potable water, which is the drinkable water, is actually taken from this groundwater. So if there is a contamination of this groundwater, you can be sure that the water which you are getting at your home is also likely to be contaminated. Number one. Then there is also a continuous, what is called as hydrologic or water interchange between the surface water and the subsurface water system. So therefore, the groundwater contamination can also move into the surface waters. Okay, so that is why it becomes quite a bit of concern that you have all of these things which are now occurring, and there are large areas which are contaminated. Now, unlike the plants, microbes will provide a very good solution for addressing such kind of pollution. So when you talk about remediating, when you talk about remediation of such kind of resources, there are two aspects which need to be understood. First is how clean is clean. That is, what is the extent of tolerance which can be allowed as far as metal is concerned, or as far as some E. coli infection is concerned, because the more purity you demand, the higher the cost is going to be involved in remediation. So one has to be very careful to define how clean do you want to carry out, how clean do you really want to make this water or a soil, and whether natural microbial activities can aid this cleanup process. Will these activities be quick enough on a time scale, or whether these can be enhanced? Okay, so here we are talking about the extent of purity, and in the second one we are talking about the time scale. How much time do I have in order for remediation activity to be undertaken if I use a microbial route? So when you talk about microbial remediation, both these things, that is the extent of degradation or the extent of removal, do I want a complete removal of the pollutants, or are there certain guidelines which I can put so that I can say that, if there is arsenic, it is allowed up to certain ppm level. If there is a hydrocarbon contamination, it is accessible under ppm level, and the time scales, they need to be defined. Okay, so both these things are important. If you say I want 100% pure water, then remember that cost which is going to be incurred will be very expensive. And eventually it just may not become economical at all to do it. So this aspect is also very important. Now, so by definition, what is this bioremediation? It is the breakdown of an organic contaminant using the microbial systems. And what you are eventually aiming is that these complex organics or these pollutants, they can be used as a source of carbon by the microbial system. And this process, given the biochemistry which we understand of microorganisms, this process can be either aerobic or anaerobic. Okay, that is, either there are going to be bacteria which can carry this out in the presence of molecular oxygen, or even if oxygen is not there. For example, when you take this groundwater, when you take this contamination of the soil by leakage of the underground petroleum tanks, okay, there the oxygen is not there. So are we trying to say that we will not be able to conduct a microbial remediation process simply because oxygen is not available there? And why are you looking for oxygen? Is because you are looking for oxidation of these by oxidation, you don't have to use oxidation term, but you can use the term breakdown. Okay, and so the question is that if you want to break down these complex organics completely into carbon dioxide and water, then even in the absence of molecular oxygen, are you able to carry out that kind of a process? So do we think that there are bacteria which can carry out such kind of a breakdown under anaerobic conditions also? Okay, that is the reason why we bring both the concepts in. That is, the process of breakdown of the complex organics can be done either aerobically, which is in the presence of oxygen, or it could be under anaerobic conditions also. So is this point clear to everybody as to what is the importance of both of these?
Yes, ma'am.
All right. So now our aim in microbial remediation is what is called as mineralization. And by mineralization, what we mean is that an organic pollutant should be completely broken down into carbon dioxide and water. And since many of these contaminants are basically, hydrocarbons, which only contain carbon and hydrogen, so the end product of metabolism is going to be carbon dioxide and water, and that process is called as mineralization. Now, under aerobic conditions, you require microorganisms which can use aromatic or aromatic compounds, complex aromatic compounds or pollutants, for example, dyes or hydrocarbons, etc. It can be used as a carbon source. It also would need a nitrogen source, which you may have to supply from outside, or it also needs essential nutrients like phosphorus, sulfur, iron, etc., and it needs molecular oxygen. Okay, so when you are trying to look at strategies which are based on aerobic routes, there is a requirement of the following things. But if you want to carry this process under anaerobic conditions, then all of these things are going to stay the same, except that the terminal electron acceptor is not going to be oxygen. Okay, because under anaerobic condition oxygen is not available, but there are molecules like nitrates, sulfates, carbon dioxide, which can also act as terminal electron acceptors in the respiratory chain. Okay, so if you are going to take carbon dioxide as an electron acceptor, then methane is going to be the end product. If you are going to take nitrate, then molecular nitrogen is the end product. If you take sulfate as an electron acceptor, then elemental sulfur is going to be the end product, because sulfate is going to be reduced to elemental sulfur. All these will classify under anaerobic oxidation. So is this point clear to everybody? What do we mean by aerobic respiration and what do we mean by anaerobic respiration?
Yes, ma'am, it is clear. One thing is like you said that we will have to provide essential nutrients like
Absolutely.
in aerobic part. The fourth point is so we will have to like supply phosphorus, sulfur, iron, manganese. Isn't it that trace amounts of this is
Trace
available in the
elements.
environment?
Very true, very true. Except that phosphorus is a major element, and you may have to give a fertilizer if you are looking at or you may have to give some kind of a phosphate salt, etc., in the soil, because phosphorus is a major element. And if there is a limitation of the phosphorus in the soil, then you will have to supply it from outside in order to promote the microbial activity. All others, manganese, iron, as sulfur is also a major element. It is a constituent of amino acids and various coenzymes. So sulfates, etc., may have to be supplied in the soil outside if you are going to carry out this activity. Okay, and you have to make sure that alternate terminal electron acceptors like nitrates or sulfates, etc., are available in the soil, and both of these options are available to you. Now, the reason we say the options are available to you is because what you are going to understand in the following slides is only the biochemistry which we know. And then if you want to develop a technology for this, then you need to prepare a well-designed consortium of these microorganisms, which can be brought together, which can be added to the soil, and as a result of which this remediation process can be carried out. So you will make an artificial mixture of all of these bacteria, which could be like 4 or 5 major bacteria, which are involved in hydrocarbon degradation, or they have the necessary biochemistry to break down these hydrocarbons. So you can prepare a mixture of these microbes, put them in a polythene bag and supply them. Okay, in fact, many of these technologies, that is the way that they are sold in the market. One such technology, which is prepared by TIRI, which is the Tata Energy Research Institute located in Vasant Kunj, they have pioneered over 20 years, and they have identified the microorganisms which are capable of breaking down these hydrocarbons. So whenever there is a petrol leakage, like in the Middle East countries, where they are constantly pumping out, crude oil, etc. If there is an oil spill, or sometimes, the ship which are on the surface of the ocean bodies, these cargo ships, when they are traveling, there could be a leakage there also of if they are carrying this crude oil from one country to another. There are huge oil spills which have been reported. Now technologies have been developed whereby these microbial consortia are available in a polythene bag in kilogram quantities, and they can simply be spread over the contaminated water, and they will completely eat up oil and just convert it into carbon dioxide and water. So this way, such large oil spills can be addressed. So is that clear to everybody? But that is only because we know what kind of microorganisms, and then they can be cultivated on a large scale in a bioreactor. Like, for example, what TRI does is that they have 1,000 liter, they have 10,000 liter bioreactors where they are cultivating these microorganisms, then packaging them, centrifuging, packaging them, and they supply it in polythene bags as a consortium of microbes, and then they can be supplied globally, and that is how oil spills are addressed. It's one of the very, very successful companies. So is that clear to everybody?
Yes, ma'am, it is clear. And also, like after eating this all wireless pills, they are in wherever they are in water body it is. So, like after that, isn't it like what will these microbes will do?
Nothing.
Like,
The microbes will simply, they will be there, and if they only use hydrocarbon, then over a period of time their life cycle also gets over, and then they are no longer present there. So these are, microbes will just decompose naturally in the water bodies. Once they have eaten up all the oil, then they are not able to reproduce anymore, and they just stay like that, and they don't bother everybody over a period of time. They are all like, the carbon, the hydrogen, and everything, they will decompose by viruses which are in the water body or whatever, and eventually they are completely mineralized to carbon dioxide and water. So they are not going to stay in those water bodies after they have done their job.
yes, ma'am.
And that is what is the best part about using either microbes or enzymes, because they are self decomposing systems. So now, the compounds, so let's see what kind of compounds can be degraded through microbial remediation. Firstly, the compounds which are very similar to natural compounds are normally completely degraded. Now, for example, there are certain hydrocarbons, etc., which are aromatic compounds, which are very similar in structure to lignin, which is already a natural compound. Okay, by microbes, what we mean, like when you see, when the plant litter accumulates in the soil, the leaves are constantly being shed, and the tree trunk, for example, is constantly shed, and it just falls on the surface of the soil. So that plant contains, the cell walls will contain lignin, and there are already microbes which are present in the soil, both bacteria as well as fungi, which are able to degrade this lignin, cellulose, hemicellulose. So today we have an understanding of what kind of microbes are there which are carrying out lignin degrading activity. And since the hydrocarbons are aromatic compounds, structurally they are very similar to the naturally occurring lignin. So the microbes which have these lignin degrading enzymes are capable of breaking down these complex aromatics. Okay, so in other words, the point which I'm trying to make here is that those particular hydrocarbons, which are structurally very similar to naturally occurring compounds, are completely degraded to carbon dioxide and water. And this occurs through participation of various enzymes. What kind of enzymes are these? These are basically enzymes which are already occurring in the cell, and they are a part of lignin cell wall, building machinery. Or partial degradation can also occur due to lack of complete set of enzymes. So you could have a hydrocarbon, but then, it may not be completely mineralized to carbon dioxide and water, simply because the microbes in that particular environment, they lack a complete set of enzymes. So there could only be a partial degradation. Okay, so it can be metabolized, but maybe some simpler compounds are there which are less toxic compared to the original compound. Even that is good enough for you, because then at least the toxicity of the compound is removed. Or you can have co-metabolism. I will explain as to what co-metabolism is. Or for example, sometimes there can be synthesis of compounds which are more complex and stable. What exactly do we mean by that? Now, for instance, there could be these dyes which are a part of textile effluent. Many of the lignin degrading compounds, they act on these dyes. However, very reactive radicals are released because many of the lignin degrading enzymes are oxidoreductases. Okay, so they generate very reactive radicals out of these dye molecules. So those reactive radicals can now, they become highly reactive, and they start to self-assemble, they start to self-polymerize, as a result of which more complex compounds can be formed compared to what you had originally in the soil. So that could also be a negative part, that the synthesized compounds are more complex, they could be more stable. That is, they are very recalcitrant, and they can stay in the soil for a long period of time. So I hope all of these points are clear to everybody. So either you have the option that microbes can bring about a complete degradation, or there could be partial degradation, which could result in so many of these activities. So this last point, that is synthesis of compounds, is explained in the last point here. Now, I have used a term co-metabolism. Let me explain what co-metabolism is. Now, co-metabolism is an activity whereby a naturally occurring enzyme acts on a substrate, which is an unnatural substrate, and converts it into an intermediate. Okay, for example, a commonly occurring enzyme in many of these bacteria which break down hydrocarbon, because there are methanotrophs, which are microorganisms which can use methane as a carbon source. Okay, so they are naturally capable of using methane. So now, uh, such microorganisms, they have an enzyme which is called as a monooxygenase. So if they are going to degrade methane, they will contain methane monooxygenase. If they can use propane, they will have an enzyme which is called as propane oxygenase. Okay. Now the interesting part is that many of these enzymes are bound, or they are cell wall bound, or they could be extracellular. And while their primary function is to act on propane, they can also act on some other contaminating molecules, like for example trichloroethylene. And trichloroethylene is one of the major pollutants. It is discharged from the dry cleaning industry or from petroleum industries as a byproduct. Okay, so this particular enzyme inadvertent, it is not designed in the nature to act on trichloroethylene, but it can act on it, it can act oxygen to it, and then it can give epoxide of TCE. And such a compound can then be acted upon by several other bacteria, eventually into carbon dioxide. Okay, so when this propane monooxygenase is acting on propane, it is called as simple metabolism. But when this enzyme acts on something which it was not naturally designed to act, it is called as co-metabolism. So are these 2 points clear to everybody? How do you distinguish between metabolism and co-metabolism? So is this point clear to everybody?
Yes, ma'am.
So for some of the factors which are extremely important for microbial bioremediation are here. And there are three factors which are important. First is the structure of the compound, number two, the toxicity of the compound, and number three, bioavailability of the compound. What do we mean by structure? That is, structurally, these organic pollutants should resemble the natural compounds. Now, as I gave you an example earlier, that lignin, which is a complex organic polymer, can be expected to induce enzymes which can break down organic contaminants. Okay, because structurally, many of these organic pollutants are very similar to lignin, and since lignin is capable of inducing some of the very key enzymes, the same set of enzymes can also act on the other aromatic compounds. Okay, so structure is going to be extremely important. In addition to that, the organic pollutants which you are looking at could exert a steric effect, or it could affect, it could have a structure which is electronically such that it cannot be easily acted upon by enzymes. So those kind of molecules will not be acted upon by the enzymes. So both of these factors are important. And number two,
Ma'am,
yeah, go on.
ma'am, lignin is like a complex polymer. So polymers can also induce enzymes. Like enzymes are induced by small molecules, prosthetic groups, and there are small molecules too.
Yeah, certainly polymers can. And one of the things which needs to be understood here, for example, look at cellulose. Okay, there are microbes which are able to break down cellulose, and cellulose is extremely long. It is water insoluble. It is present outside of the cells. In such a scenario, the enzymes constitutively produce cellulases. There is a leaky expression of those enzymes, and these enzymes, since they are extracellular, they are secreted out of the biological system, out of the microbes. And cellulase will act on this constitutively produced cellulase will act on cellulose to release molecules like cellobiose or other disaccharides, for example, which are now capable of getting back inside the cell. Because these are soluble compounds, they enter inside the cell and then they bring about induction of the cellulase genes. And the same thing happens in the case of these other oxidoreductases. So the polymer could be very, very complex. Okay, like lignin is a polymer, and but there are lacases which are produced constitutively, which are extracellular enzymes. So extracellular lacases will go outside. They will break down this lignin molecule into smaller molecule like veratryl alcohol, or it could be propyl alcohol, which are small molecules which enter inside the cell. They are the real inducers of the lac operon. Okay, so in this way,
Yes, ma'am.
even complex polymers, that is the way the bacteria and fungi which break down lignin, like for example, the leaf is there, but gradually over a period of time, you will see that there are soil bacteria, the soil fungi, which have completely broken down the leaf. You don't see anything there. Everything is mineralized to carbon dioxide and water, including the lignin, the cellulose, the hemicellulose, the pectin, and so many other complex polymers which are present in the plant leaf. Okay, so that is the first point. Structurally, they should be similar, because so that they can induce those same set of enzymes, and that is how the remediation can occur. And the second point is that a period of adaptation or acclimatization is usually necessary when you talk about microbial remediation. Okay, now that means that so once there is a new organic pollutant which enters into the soil, okay, the microorganisms there may not be able to synthesize those enzymes, but rather it could take time. It could take several weeks, for example, for them to get acclimatized. And during this process, okay, how the biological system prepares itself is that either there is a mutation which could occur, because mutations are spontaneously occurring in nature. So if there is a spontaneous mutation which occurs in this microbe, as a result of which one of its enzymes is modified in such a way that its substrate specificity now increases, so now it can also act on that organic pollutant. The same enzyme has now a very broad substrate specificity, and it can act on that other, which is slightly modified, aromatic compound, or a gene transfer, because there are other kind of bacteria which are present in the soil, and through conjugation and other things, one of the organisms which was very good in hydrocarbon degrading, a plasmid gets transferred to it from something which can also break down the textile dyes. So this particular bacterium now becomes very versatile. It can break down hydrocarbon, it can break down textile dyes, and it can act on that soil much more efficiently. Okay, and this can take a long time. So in other words, acclimatization, when you think about microbial remediation, acclimatization is also extremely important. Okay, so I hope this point is clear to everybody, that what do we really mean by adaptation, acclimatization? Why is it necessary? Why are the structures of the organic pollutants necessary? Number two, the second factor is toxicity. Now, if you have, for example, these chemical spills which are occurring, or there are engineered remediation projects, I will just show you in the next slide as to what we mean by that, which is called as land farming, etc. They can sometimes have very high concentration of the contaminant, as a result of which toxicity can occur, and it can slow down the remediation projects. Okay, or the contaminant may get into the cell membrane, disrupting it, in which case, for example, again, some time, acclimatization, etc. has to be given for microbial remediation to be successful. Okay, now what do we mean by land farming? Now, here is what is land farming. Okay, so land farming is a bioremediation technology, and it has been successfully operating since 1980s. It is primarily used for treating petroleum contaminated soils or sediments. Okay, so the material which is to be treated, so there could be soil, which could be an underground soil, because there has been a leakage of these storage tanks. So large cranes, they will bring, tons and tons of these soils or sediments. So the material to be treated now is incorporated into the surface soil. For example, you see here, so it is spread over many acres of land. This contaminated soil is mixed, and it is just kept with the natural soil, and it is amended with nutrients, naturally occurring microbes in the soil, or you could have a consortium which is added. They are going to transform the waste materials, the organic contaminants, to carbon dioxide, water, and biomass. Okay, so eventually, over a period of 3 months or 4 months, the soil is completely clean. All the organic pollutants have been removed, and this soil can go back to wherever it was. Okay, so such projects are called as land farming projects, and they have been, for the last 40 years, like you see here. It's constantly going, but then of course, there is a lot of it needs to be mixed with some soil in a certain proportion, because otherwise the levels of toxins is so high that a microbial activity cannot be supported in there. So you need to mix it with the normal soil. That's a normal practice. Okay, so I hope this point is clear as to the point of toxicity. Or number two, there are some functional groups which can be present. For example, you could have, let's say, if it is a simple benzene ring, there are, or it could be just benzoic acid, which can be broken down by, there are microbes which can easily break down benzoic acid also. That can be taken up. They can use it as a carbon source. However, if there are halogens which are added in there, okay, like the trichloro benzoic acid, the dichlorobenzoic acid, etc. Okay, now addition of these halogens can make it very, very toxic. Okay, so that's what we need to understand, is that in spite of that, the organic pollutants could be containing certain kind of functional groups, which makes them particularly more toxic. Okay, so these physicochemical properties of the contaminants need to be understood very well, so that one can address, or one can use microbial remediation as a technique. Now, so is this point clear to everybody as to what we mean by both structure as well as toxicity?
Yes, ma'am.
Yes, ma'am.
Okay, so All right. So, and the third point is bioavailability. By bioavailability, what we mean is that it's true that, you have a microbial consortium, you have added it to the soil, but is it going to be taken up inside? Because unless it is taken up, especially if these are small molecules and if there are no extracellular enzymes to break them up, are they available to the microorganisms? So bioavailability is another important factor for microbial bioremediation to occur. So there are four ways that the hydrocarbons can be taken up by the microorganisms, which are shown in this picture, and all the points are explained here. So the first is that utilization of the solubilized organic compound. Okay, like you see here, for example, so the blue thing represents it's a bacterium, and you see all of these here, these are the flagella, because in order to migrate through water, the bacteria also have these filamentous structures which are called as flagella. Okay, so either because many of these, whether it is methane, whether it is propane or whatever, they do have limited solubility in water. So if they are solubilized in water, then the microbes can directly take them up from the aqueous phase. Okay, that's what this point means, that utilization of the solubilized organic compound. Okay, or for example, the uptake could be by making a direct contact of the cells, but via these fimbriae or via these dots. So what you see here is, for example, that the hydrocarbon, it could be like oil, and oil droplets will all combine together to give you this hydrocarbon phase. So the microorganisms can become attached to this. Okay, and then so via surface modifications or whatever, so they will directly become attached to the hydrocarbon surface, then they can take up these molecules inside the cells. Okay, it is not really the solubilized one like you saw here, for example, but the microbes become attached to the hydrophobic phase here. The third one is that the microbes can directly make a contact with either fine or sub-millimeter size oil droplets, like in this particular case. Okay, so some droplets are there which is not really, which is detached from the cell surface. The microbes can also make contact with these fine droplets, etc., and that they can take them inside. Or finally, biosurfactants or emulsifiers, like no, in C, you had the direct contact in this particular case, and D is actually a biosurfactant or an emulsifier. So many of the microbes, they also make these emulsifiers or biosurfactants. So what they do is that these are like missiles to which both the microorganism as well as the oil droplets get attached to these missiles, which allows these microorganisms to make an intimate contact with these oil drop with the oil droplets or with the hydrocarbon molecules. So there are, in principle, for bioavailability, all of these four principles are operating, and depending on what kind of microorganism you're going to select, for example, so you may have to, if you're going to make a consortium, you may also have to, for example, mix it with some kind of a biosurfactant producing bacterium. Okay, for example, if these consortium have to be added to a water body, then a surfactant also needs to be mixed. Okay, so that's the reason why attention needs to be paid to such kind of details.
Ma'am, can you please explain again what are how the surfactants are being produced?
Okay, so well, I also have a slide which I have just come to now. So these biosurfactants or emulsifiers, what they do, that they increase the solubility of the hydrocarbon by making micelles in the water body. Okay, so micelles, these are structures which have both hydrophilic part and the hydrophobic part. Okay, to the
Yes, ma'am.
hydrophilic part, the cells will get attached, and to the hydrophobic part, the hydrocarbons are going to get attached. So in essence, they increase the solubility of the hydrocarbon. Okay, and as I said, that they do facilitate attachment of the cells to the hydrocarbon by making the cell surface more hydrophobic, and therefore they are better able to stick to the oil phase. Okay, so when microbial consortia are designed for treatment of the hydrocarbons, they make up important components of the same. That is the biosurfactants, etc. And There are non-biological surfactants also. Okay, like twin 20, twin 80, or triton X100, but they are all chemical surfactants. And once you add them to the microbial consortium, these are going to be left behind. Okay, and they will also add to the contaminant load. But when you say a biosurfactant like octyl glucoside, like hexyl glucoside, so all of these are called as biological surfactants. First, they are produced by microbial systems, and number two, they are self-degrading. So something like an octyl beta D glucoside contains an octanol chain, and through ether linkage there is a sugar which is attached to it. That is, remember what is octanol. Octanol is a long hydrophobic chain, and there is a hydroxyl group. Now it can bind to another sugar molecule. The sugar also has hydroxyl groups. So the hydroxyl of the sugar and hydroxyl of the octanol will combine in an ether linkage to give rise to octyl beta D glucoside. That is a biological surfactant because it has the sugar and it also has the hydrophobic component. So they assume the structure of a missile. The advantage is that these biological surfactants can be degraded naturally in the water bodies through other kind of microbes which are occurring. They will use them as a source of carbon. Okay, so therefore the advantage is, and that's the reason, I could have just used a term like a surfactant or an emulsifier, but the reason we are using the term biosurfactant is because it is a surfactant which is produced by a biological system. It is self-decomposing. So when you add it to the microbial consortium, when you are developing, or when these commercially available consortia are available, like oil zapper, I'll give you an example of that. They all contain these biosurfactants also. So if this is clear to everybody, let me move on and tell you something about now when we talk about microbial remediation, what kind of contaminants are suitable for bioremediation. There is a long list of these. For example, you see the first one is the chlorinated solvents. It has been found that this is a candidate contaminant which can be degraded because in several microbes there are enzymatic activities which can break down the chlorinated solvents, or polychlorinated biphenyls such as 4-chlorobiphenyl, 4,4-dichlorobiphenyl. Where do they come from? These two compounds, they come from electrical manufacturing, power stations. They also come from the railway yards. Similarly, chlorinated phenol, like pentachlorophenol, comes from timber treatment. That is, when wood is processed, the timber is treated with certain solvents during polishing and other things. So during timber treatment or during land refills, all of these organic pollutants, or like look at chlorinated solvents, they are used for dry cleaning purposes. So where is all that going? After that solvent is used up, it is simply discarded outside. They just fill it up in the buckets and they just discharge it anywhere. So is that clear? So similarly, the BTX compound, I had told you that BTEX is collectively used to define mixture of benzene, toluene, ethyl benzene, and xylene. And these BTEX compounds are released from oil production and storage, oil storage, that is from gasoline tanks, gas work sites, from airports, because many of these airports, they also have all of these gasoline storage tanks which are filled into the aircrafts, or paint manufacture at the shipyards, port facilities, railway yards, chemical manufacturing processes, so on and so forth. Okay, polyaromatic hydrocarbons like naphthalene, anthracene, etc. And you see what is the source of all of these compounds. So you see a long list of these organic contaminants. Okay, but they can be removed through microbial bioremediation. Why? Because structurally these are mostly aromatic compounds, and microorganisms have the capability of degrading such kind of aromatics. In addition, there are various aliphatic hydrocarbons, which are also released into the environment, and they can also be attacked by microbes. By aliphatics, what do we mean by simple linear aliphatics? Okay, like methane, propane, butane. All of these are not aromatic compounds, they are aliphatic hydrocarbons. Okay, and they can also be attacked by microbes because there are methanotrophs which can use methane, they can also use propane and so many other hydrocarbons. Okay, and the third reason is that there are a limited number of enzyme systems which are present in the microbe, okay, which are capable of degrading these into intermediates which can then enter into the TCA cycle. Okay, that is the reason why we say that microbial remediation is a process which can successfully be applied in order to address the hydrocarbon contaminant, either aliphatic or aromatics. Okay, so I'm just going to very briefly look at, just explain to you, give an example of like benzene, toluene, or naphthalene, etc. What kind of activities are present in the microbes which are capable of degrading them. So is this point clear to everybody?
Yes, ma'am.
Should we move quickly forward? Okay. All right. So crude oil is an example which I'm going to consider, and I will tell you that all of these things which we talked about, the organic pollutant and whatever, they actually come from crude oil. So during the processing of the crude oil, when you make petrol, okay, there are pollutants which are called as the saturated pollutants. And what are these? These are aliphatic. They can be either linear or cyclic. That is, what are these saturated compounds which are released as a byproduct of petroleum industry? These are hydrocarbons, and since we say saturates, these are without double bonds. Okay, and they represent the highest percentage of the crude oil constituents. Then there are aromatic compounds, which are the ringed hydrocarbons. Okay, the crude oil also contains mono rings like you have in benzene, or they may contain several rings like you have in naphthalene. There could be substitutions in the ring like methyl substitution. For example, look at toluene. Toluene is basically a benzene ring, but then it contains a methyl group. Or there could be phenolic compound, which is a benzene ring, but which has now a hydroxyl group. That's what we mean by substitutions in the ring. Like toluene, ethylbenzene, or xylene, etc. Xylene has two methyl groups. Okay, and collectively, all of these are called as the BTEX compounds. Okay, in addition to this, the crude oil also contains resins and asphaltenes. What are the resins which are present? These are basically solids which are dissolved in the oil, and they are non-hydrocarbon compounds which also contain. So both of these, the aromatics, the saturated that we talked about, they are only hydrocarbons. They contain hydrogen and carbon, but resins are non-hydrocarbon, and they also contain nitrogen, sulfur, and oxygen. And that is the reason when diesel oil is burned or when petrol is burned, if it was only hydrocarbon, then only carbon dioxide and water would be released. But since your petrol also contains the resins and the asphaltene is also there, quite a bit of it, that is why oxides of nitrogen like nitric oxide, nitrogen dioxide, or oxides of sulfur are released. Is that point clear to everybody?
Ma'am, can you explain what are asphaltenes again?
Okay, so the crude oil contains, apart from these uh, aliphatic compounds, which are basically linear compounds, they can be cyclic also, like cyclopentane. That is completely saturated. Okay, it doesn't have any double bond, but it's a cyclic compound. All of them are clubbed together in the saturates. Then you can have aromatic hydrocarbons, and all of these are only hydrocarbons. Okay. Apart from that, the crude oil also contains resins and asphaltenes. So what are resins and asphaltenes? These are non-hydrocarbon constituents of the crude compound. They are polar compounds. They are not non-polar. They are polar compounds because they also contain nitrogen, sulfur, and oxygen. Oxygen, oxygen can give capability of, or phenolic compounds, they can give, they lend solubility to these aromatic molecules. They have, so the resins and asphaltenes, they have a unique chemical composition that affect their biodegradability. The resins will contain aromatic compounds which have very long alkyl chain. They are soluble in N-heptane and N-pentane. They are structurally very similar to surface active molecules in the crude oil, and they act as peptizing agents. What is a peptizing agent? So these are materials which, when added in relatively small amounts, are capable of causing substantial reduction in the viscosity as a result of depolymerization or dispersion. And peptization can occur in aqueous medium, such as when agents are added to prevent flocculation or aggregation of particles. Okay, so please try to read it and try to understand. So resins are aromatic compounds, except that they are soluble in long chain hydrocarbons like heptane and pentane, and they are very structurally similar to surface active molecules which are present in crude oil. Okay, and they act as peptizing agents because, in other words, they they cause a reduction in the viscosity, resins, etc., and they are colloidally dispersed in the saturates and aromatics. So it is like a colloidal suspension in the crude oil. That is what is called as resin. Okay, then these are viscous, high molecular weight compounds containing polycyclic clusters, etc. And these are, resistant to biodegradation. The peptizing agents such as resins, they keep asphaltenes in suspension, thereby promoting the stability of crude oil. Okay, so I hope it is clear to everybody that in the crude oil, for example, you have saturated compounds, you have the aromatic compound, you have asphaltenes, you have certain resins, etc. So whenever there is a leakage of crude oil, remember, leakage can be of the petrol, but leakage can be of the crude oil also, in which case the soil will get contaminated with resins and asphaltenes, etc. In fact, Dr. Preeti in our department, she works on asphaltene degradation. So she has a unique consortium of microorganisms which can break down asphaltenes. And she's collaborating with Indian Oil Corporation, where they are using some of these microbes which are developed in the department for bioremediation of asphaltene, which appears as a leaked compound. Okay, so is this clear to everybody? Is this clear
Yes,
to
ma'am.
everybody? Please try to read it. It's very, very interesting and very fascinating when you talk about crude oil, what all it contains, what kind of biochemistries you need to learn in order to develop the technologies for their, for their removal. Yeah, okay.
There was a paper
So,
published by Preeti ma'am last year
yeah,
about,
yeah,
I think,
exactly.
yeah.
Asphaltene. I'm also a co-author because
Yeah,
we have
you were.
collaborated. Yes, yes. So,
Okay.
so no, of course, she has moved, extensively now into that area, asphaltene. So, you have an overview now of what all does crude oil contain. So, let me give you what kind of biochemistries have been found in nature in microbial systems. Firstly, both aliphatics and aromatics. Now, aliphatics are non-aromatic compounds. Okay, because first of all, since they don't have any double bond, aliphatic can be both linear as well as it can be cyclic, like cyclopentane. Cyclopentane is not called as an aromatic compound, but because it doesn't have any smell. Basically, that is where it is derived from. All aromatic compounds like benzene, etc., they all have a certain smell because of this presence of double bond, conjugated double bond system, and which gives it very special chemical property, and that is why these compounds are called as aromatics. Okay, so the phenol, the xylene, the toluene, the naphthalenes, etc., all have smell. And so structurally as well as in their physical property, they all smell. So they are called as aromatics. Okay, there are both aerobic roots as well as anaerobic roots which have been found in microorganisms. Under aerobic roots, okay, both aliphatic hydrocarbon as well as aromatics can be broken down. Okay, so let me just I have Um, I'm just going to put the slide here so that you can see All right, so I have come back. So can you see the slide here? Degradation of aliphatic and aromatics.
Yes, ma'am. Yes.
Okay, all right. So let me go to the full PPT mode. And so as I was saying, under aerobic route, both aliphatic as well as aromatic hydrocarbon can be degraded. Even under anaerobic conditions, both aliphatic and aromatic hydrocarbons can be broken down. And, so I'm going to explain these, but as an overview, I want to tell you that the aliphatic hydrocarbons can be acted upon by either monooxygenases or dioxygenases. So enzymes which are capable of adding a single atom of oxygen or two atoms of oxygen to the hydrocarbon. Okay. And the process can be carried out either by what is called as terminal oxidation or subterminal oxidation, which I will explain in the next slide. And further metabolism results in the formation of a primary alcohol, aldehyde carboxylic acid, and it is then converted into an acyl coenzyme A. And such acyl coenzyme A, okay, can be degraded by one of the very well established pathways which occurs in all cells, which is called as a beta oxidation pathway. Okay, and beta oxidation pathway converts long chain fatty acids, like you can have propanoic acid. Well, propane is very small, but in the hydrocarbons, you can have long chains like C10, C12, and their corresponding acids are made. Okay, and then they break down that into acetyl coenzyme A, which then enters into the TCA cycle. So this way, the aliphatic hydrocarbon can be. So is my voice still clear?
Yes, ma'am.
Okay, all right. I thought maybe there was some disconnection or something. So okay. Now, the same thing, for example, by aromatic hydrocarbons can be degraded by certain pathways, and they're very easy to understand. There are only just very limited ways that they can be done, which I will explain in the next slide. Under anaerobic routes, for example, since molecular oxygen is not there, okay, so what they carry out are basically three different pathways, which are called as either unusual oxygenation, about which we don't know much, or there is addition of, like it says, carboxylation. So carbon dioxide is added to these molecules, or a fumarate is added by these group of enzymes. Okay, so whether these are aliphatic or aromatic, they are going to be broken down exactly by the similar kind of mechanisms. So let me explain right here under aerobic route what happens. Okay, so very quickly, this is exactly from the same slide which I showed you in the last slide. So we have either mono or dry oxygenation which is occurring. Okay, so I had told you that either terminal or subterminal oxidation can occur, and this is explained here. Okay, what do we mean by terminal oxidation here? For example, here is a propane molecule, okay, and the enzyme alkane hydroxylase, it carries out terminal oxidation. That means it is the terminal methyl group, which will be now to which an oxygen is added, and the enzyme here is called as alkane hydroxylase. Okay, they're also called as monooxygenases. Because you see, there is no difference here, except that there is a single oxygen which is added. Okay, and this is what is called as a primary alcohol, and that is what is written on this side. So a primary alcohol is made simply by addition of a single oxygen by a monooxygenase. In the next step, alcohol dehydrogenase will act on it in order to give rise to an aldehyde. Okay, and then it is oxidized to give rise to a carboxylic acid. And then acetyl coenzyme A synthetase will add a coenzyme to it. Okay, such kind of moieties, they will undergo beta oxidation. In a beta oxidation, you will remove one moiety here. If it is a long chain hydrocarbon, then two moieties are released. That is, acetyl coenzyme A is periodically released in order to give rise to acetyl coenzyme A. And that's what you see here. And that is how it enters into the TCA cycle, and it gets completely metabolized into carbon dioxide and water. So is this clear to everybody? Can you see that how different enzymes, for example, can bring about oxidation of this hydrocarbon, which is aliphatic hydrocarbon? Okay, the second pathway is the subterminal oxidation. Like I wrote here, the subterminal oxidation. That means it is not the terminal methyl group, but the subterminal methyl group to which oxygen is going to be added. Otherwise, the chemistry is exactly the same. The same group of enzymes, in other words, they have a broad substrate specificity. They can add oxygen either to the terminal carbon or to the subterminal carbon. And, like here, it is this particular methyl group. Same set of enzymes, except that once you make this, the second compound which is going to be formed is a ketone. Then you have an ester, and then esterase will come in order to break down the acetyl linkage, and you have acetyl coenzyme A, which enters into the TCA cycle. And the primary alcohol which is made now, can get into this cycle, and it can eventually be metabolized. Okay, so I think it should make it very, very clear to you as to why, for example, as to how the aliphatic hydrocarbons can be degraded by microbial systems, because they contain all of these necessary enzymes. And in addition to that, I also said dioxygenases. So the dioxygenases, they carry out this kind of a reaction. Alkane, they add two oxygens. The resulting molecule is called as a hydroperoxide. That is converted into a primary alcohol, into aldehyde, into fatty acid, and eventually the fatty acid is converted into this fatty acyl coenzyme A. It undergoes beta oxidation, and then it enters into the TCA cycle. The second point which is important here is that such kind of enzymes, although I have just shown it, for propane or whatever, but many of these enzymes which are in these remediating bacteria, they prefer C10 to C18, because remember, the lower alkanes, the lower hydrocarbons are all gases. The methane, the propane, the butane, they are all gases. But the higher hydrocarbons, starting from C10 to C18, for example, there is an increased solubility of these. They are more soluble in water, and that is why they can be degraded more easily by the microbial systems. Okay, and the branching, the number and the position of where the methyl is going to be located, methyl group, like in the case of toluene, or you have xylene, etc. That is also very, very important when you talk about microbial bioremediation. Okay, so is this point clear to everybody? Have you been able to understand? So please pay attention to it. It's not very difficult to understand. It's a very simple series of reactions. So is that okay? Is that understood?
Yes.
Okay. So now, we had only looked at the aliphatic hydrocarbons, but in this slide, I'm showing you the aromatic hydrocarbons. Okay. And these are what are extremely important, and they have been listed as 16 priority pollutants by the US Environmental Protection Agency, which is one of the biggest governing bodies who sets up the standards for organic pollutants, etc. And if you look at all of these, like naphthalene, anthracene, phenanthracene, they can contain either 2 aromatic rings or 3 or 4 or 5 multiple of these aromatic rings, or benzopyrene, for example, or this. Okay, and they are all aromatic compounds because you see that there are double bonds in their structures. What is the source of these pollutants? Incomplete combustion of organic material like coal, tar, oil, and gas, automobile exhaust, tobacco burning, or smoked food, either during industrial activity or other man-made activities. All of these are released during these processes. Okay, and they are also released as a consequence of geothermal reactions, which are associated with the production of fossil fuels and minerals, and they result in all these polyaromatic hydrocarbon formation. Okay, so currently, before the microbiological roots came into picture, there are several physical and chemical treatments which have also been looked at. It's not like people have not been looking at how to remove these, but mostly these methods have been either incineration, like if you have some naphthalene or something else, people think let's burn it out as much as possible, or there is base catalyzed dechlorination, which is a chemical treatment, or UV oxidation, or UV fixation, or solvent extraction, etc. Okay, so people have tried many of these methods, which are physical and chemical treatments, but the drawbacks are that they are extremely expensive, complex to carry out. They carry with them the regulatory burden because they don't, they also result in accumulation of several other things which are equally bad. Also, they do not destroy the contaminated compounds, but they instead transfer them from one environment to another. That is, either it could be in a solid environment and it is changed into a liquid environment, or you could be changing them from one phase to another, from aquatic phase or from organic phase into aquatic phase. Okay, so please try to understand the chemical treatments are there, but they have huge drawbacks which are associated with them. And since microbes, as you saw, they can take up these compounds and completely degrade them to carbon dioxide and water. So there is no load as such. So is this clear?
Yes, ma'am.
Okay. So now I think let me stop right here. We are just 4 minutes short, and so just read up to the last slide, for it'll be a part of your quiz also. And I will just upload the PPT. So the remaining part of microbial remediation, I'll take it up in the Monday class. Okay. So we have
Okay,
quiz
ma'am.
only for half an hour. Okay then. Okay.
Yes, ma'am. Thank you, ma'am.
Okay. Okay. Fine. And if there is anybody who has a question, you can even post it. If you want tomorrow, maybe tomorrow in the evening around 7 o'clock, I can take a half-hour doubt clearing session for you. If you quickly want to revise something with me, it's okay. Just send a post to me.
Yes, ma'am. That will be better. Like in evening, if you can organize a doubt session, that will be good.
Certainly, you talk to each other on the Teams, and if you want half an hour or 1 hour with me tomorrow evening, I can sit down with you and let me know, and we can fix it accordingly.
Yes, ma'am. Yes.
Okay, okay.